Bismillahirrahmanirrahim. Students, today we are going to discuss about how the characters they are transmitted from one generation to the next generation and how they are segregated according to the law of segregation, which was derived by Gregor Mendel. Okay, you have stated this something in your lower classes that Gregor Mendel was a priest. Okay, and he had the hobby to grow different types of the plants. Now look at his his keenness in the pattern of transmission of the characters from one generation to the next generation. He did grow the the Pisum sativum. What? Pisum sativum. And he observed that there were certain plants which were tall and the others were dwarf. Okay, so this is tall and this is dwarf. When he crossed these two plants, he took the pollens from the tall and applied those pollen on the stigma of the dwarf plant. He found the result that all the plants which were grown from the seeds taken from formed from the combination of these two plants, they grew as the tall plants. Okay, he called them as first filial generation or F1 generation. What? First filial generation or F1 generation. Now Mendel self-grasped these plants. Self-grasp means what? Say here is suppose these are the pollens, okay, and these are petals, okay. We are at the moment not concerned with the petals. So this is say for example stigma. He took the pollens from the same plant and allowed them to have the chance of fertilization. Okay, so this is what the phenomenon or the procedure which was used by him. He used the pollens of the same plant, plant of first filial generation, and used them on the stigma of the same plant. This is known as self-fertilization or self-cross. He took the seeds and then grew them, and he found that about 27% of those plants which were grown after grown from these seeds, he called them what? So suppose this is the seed, okay, and he grew those seeds, okay. These are the F1 seeds. Now these are the F2 generation plants. These plants and these plants. He came to know that in the F2 generation, the tall and the dwarf character, they are segregated, they are separated. So he called this phenomenon as segregation. Okay, he called these tall plants as the hybrids. What? Hybrids. He just visualized it was his intuition. At that time, I will tell you people that in 1855 and 56 and 57, there was no more concept of genes or DNA or chromosomes. Chromosomes were discovered in 1876 by Waldea. Okay, and then were confirmed by another scientist. Okay, uh, by Fleming in 1881 and 82. Okay, well, so there was no more concept of genes. He called certain thing which controlled the dwarfness and tallness of the plants, and he called them elementon. What? Elementon. Okay. Well, gene was used. The term gene was used afterwards. Okay. Well, he said that there is. Now, I will tell you people about his visualization or his interpretation. He said that this tall plant and this dwarf plant is actually controlled by a pair of a pair of alleles. Okay, and he said that these are dominant, and he said that these are dwarfs. That very character which appeared in the hybrid condition or in F1 generation, he said, he termed that, he dubbed that character as the dominant one. So he said that the dominant character was controlled by, he just supposed that was controlled by a pair of dominant alleles. And he said that the dwarf character and the trait was controlled by the by what the recessive traits are recessive elementons. Okay, a pair of recessive elementons. Now he said that in the gametes, half of the elementons are transmitted. Okay, and he called this something as genotype, and this is what the phenotype. We are discussing what the interpretation of this experiment. Okay, now in F1 generation, this one, he said that the tall elementon, they dominated the dwarf ones. Okay, and the genotype should be capital T, small t. He said afterwards that capital T, small t, when they are self-crossed like this, what happens? He said that this is the gamete, this is the gamete. He said that this could be what? The female. Okay, this could be female. This could be what? The male. Okay, this could be male and this could be male. Okay, these are the chances. And he came to know, he developed this this possibility. Okay, I will tell you people that this square was developed by William Punnett. Okay, so it is called Punnett square. Punnett square. Okay, and before that he said what capital T and small t, capital elementon and small capital elementon and capital elementon should have the chance to unite together in the next generation, and same chance could be there. Capital T should have the chance to combine with the small t, and the genotype would be this. Similarly, this one and small t, small t. Now you see this 25% and this 25% makes what? 50%. These are this 50% and this. If you round up, what it makes? What? 75%. 75% tall means 25% pure capital T, and 50% capital T, small t. But how would we come to know that the plant which is in F2 generation, either it is capital T or capital T, small t? So this was he devised a test, or what, an experiment, or the second part of the experiment, to deduce, to come to know either the plant was having what, the capital T genotype or capital T, small t genotype, okay, or hybrid or pure, okay, or homozygous or heterozygous, okay. So he crossed to know either this plant, either this very plant of F2 generation, this very plant, is capital T or capital T, small t. It is pure, I mean homozygous dominant or heterozygous dominant. He devised a test, and that test was what he that was called as what, test cross. What, he crossed the plant with the recessive plant, small t, recessive of F2 generation, are the pure type. Okay, what happened if it were what, capital T, then all of the I should say F3 generation should be what, capital T, small t, means all tall, 100% would be tall. Okay, and if this is the condition, what will happen? Capital T, small t. Look, capital T, small t multiplied by what? Small t. What will happen? Capital T, small t, and here could be the chance of what? Capital T, small t. Here is always small t. What will happen? Capital T, small t, and small t. Means what? 50% of, if the 50% of the next generation is tall and 50% is dwarf, then the plant which you have, that would be what? That would be heterozygous. So this is what called the test cross, the cross between homozygous recessive and a hybrid, or what, the plant of F1 generation is known as what? Test cross. Okay, I think you should have understood, but I may translate it in the Urdu language. उसने ऐसे किया था, टेस्ट क्रास में टेस्ट किया था कि जो फिनोटाइप है, जो डोमिनेंट फिनोटाइप है, वो हाइब्रिड है या प्योर है, वो होमोजाइगस है या हेटेरोजाइगस है. तो उसने क्रॉस किससे करवाया था? उसके साथ करवाया जो कंफर्म, जो फिनोटाइप, ऐसी फिनोटाइप जो जीनोटाइप और फिनोटाइप एक ही होती है, और वो क्या होती है? स्मॉल टी. ओके, यानी ड्वार्फ. उसके साथ जब क्रॉस करवाएं तो अगर तो 50% उसमें आते हैं व्हाट, डोमिनेंट, और 50% रिसेसिव आते हैं, तो इसका मतलब क्या होगा? आपके पास जो टाल है, आपके पास जो डोमिनेंट है, वो क्या है? हेटेरोजाइगस है. और अगर वो अगर उसमें 100% आते हैं, क्या टाल आते हैं, तो इसका मतलब क्या हुआ? आपके पास जो डोमिनेंट फिनोटाइप है वो होमोजाइगस है. अब एक बात रह गई, और वो ये कि क्या लॉ ऑफ सेग्रीगेशन के जरिए से, आप एनिमल, इस लॉ को एनिमल पे अप्लाई कर सकते हैं? जी बिल्कुल कर सकते हैं. आप गायें, भैंसें, बकरियां, मुर्गियां, तमाम इन जानवरों पे अप्लाई कर सकते हैं, ठीक है। उनकी F1 जनरेशन जो है उसको आप करवाते हैं, F1 के इंडिविजुअल को F1 के इंडिविजुअल के साथ क्रॉस करवाते हैं, तो F2 में वो कैरेक्टर्स सेपरेट हो जाएंगे। लास्ट क्वेश्चन, आइदर law of segregation can be tested on the human being or not. The answer is what? Not. Rather big not. Why? Because in a human being, it is not possible to have the self cross of the F1 generation. It is not possible to to have the cross between the sister and the brother. Okay, that is the reason that law of segregation cannot be applied or studied on using the human specimen. Okay, if you have understood well, it is your duty to share all this information, all this this lecture to your friends.